Good morning, everyone, and welcome to the second episode of PLASTICAST, the Pacific Northwest Consortium on Plastics podcast. I'm your host, John Dickens. I'm a master's student at Oregon State University, and I study the impact of microplastics on animal behavior and physiology. PLASTICAST is a mini-episodic series interviewing scientists on the latest in plastic and ecotoxicology research, legislators in policy development, and other interested stakeholders. In our first episode, we discussed the goals of the Pacific Northwest Consortium on Plastics and the ecological threat of microplastic pollution. Joining us today for our second episode is Dr. Susanne Brander of Oregon State University. Dr. Brander leads the research side of the consortium and also happens to be my graduate advisor. Thank you for being here with us today, Dr. Brander.
Thank you so much for having me.
So the Pacific Northwest Consortium on Plastic is composed of two parts. Last week, we learned about the consortium side, but this week, we want to hear a little bit more about the research side. Could you discuss that a little bit?
Sure. So the research side is a really exciting collaboration between several different scientists who come at this problem from different points of view. So we have Dr. Stacy Harper, who is an environmental engineer by training, who studies nanoplastics and nanoparticles. Myself, I'm an ecotoxicologist who has been studying environmental stressors like pesticides and other pollutants, including plastics, for a number of years, mostly thinking about things that are salty, estuarine or marine ecosystems. And then we are lucky to have a couple of great faculty from the Hatfield Marine Science Center here at OSU, as well as Dr. Wayne Landis from Western Washington University, who is an ecological modeler who's going to kind of tie all of our different results together. And at Hatfield, it's Dr. Chris Langdon and Dr. Matt Hawkyard, who works in his lab, and they're going to be looking at oysters. And so we've really have a unique opportunity to approach this from the Harper Lab's perspective, which is looking at freshwater organisms and responses in, say, zebrafish and Daphnia magna, which is a water flea that zebrafish prey on. And then in my lab, we're looking at an estuarine model, the inland silverside, and the mysid shrimp, which is a common model used in environmental stress studies. And we're looking at that predator-prey relationship in terms of plastics there in our lab. And then the oysters, of course, are important for several reasons. One, that they're a sensitive bioindicator. And then also when you're thinking about early life stages, they're going to be even more sensitive. And of course, it's a seafood item, which is of concern for human health and human consumption. And what makes this research unique is that we have these three different groups collecting data in the lab that using experimental designs that are informed by what's happening in the field, at least ideally. And then we have Dr. Landis coming in and using those data to really give us an informed assessment of risk, rather than just thinking like we have been so far about plastics. Well, this is probably a hazard. It seems bad that we're dumping plastic in the ocean, right? Anyone who sees that happening is, this probably isn't a good thing. But we really need to know what that means in a quantitative way to be able to tell managers and industry and all these other stakeholders that Stacy Harper had been referring to, how to go forward. What's the best plan for the future to make this better?
Thank you. So you mentioned a lot of different research projects that are underway, a lot of animals across different salinity gradients, and all sorts of different physiological research is happening. But could you discuss any areas of microplastic research where there are knowledge gaps, or you feel that are in need of the most urgent attention?
Sure. I think the biggest knowledge gaps right now are on dose response. So a lot of data have been collected in wild organisms on occurrence. This species was found to contain this much plastic. This organism had X amount of plastic trapped in its gills, or we found this type of plastic in sediment. What we don't know as much about is how organisms respond to those specific types of plastics. So, for example, tire wear particles have been found all over the San Francisco Bay. They've also been found in the Charleston Harbor off the East Coast. So we know they're there, but we don't know how much is a problem. And so one of the experiments that we're doing in the lab right now in freshwater fish, as well as in estuarine fish, and we'll be doing this in oysters as well, is exposing them to known concentrations of tire particles and then looking at things like swimming behavior, respiration, growth, even gene expression, so we can get at what's happening at the molecular level. So really, the gaps in knowledge are from just not having specific information on what amount of plastic causes a particular effect. We know it's out there, but linking it being out there to a negative consequence is what's missing. And a lot of the current literature, including things that I've published from my own lab from previous grants several years ago, tests concentrations that are at much higher than environmentally relevant concentrations. So we're doing this to get an idea of what's actually happening, considering the concentrations that we're finding in the environment, now that we have a better handle on what those concentrations are.
That's great. So obviously we're learning that plastic pollution is an increasing problem. What is one thing that our listeners could do to reduce plastic pollution in their lives?
Sure. And, right now, in the time of COVID, this can be a bit more challenging and has become a bit more challenging for my family as well. And a lot of these things take time, but things you can do are you can go on websites like zerowaste.com. There are many of them out there that will give you ideas of how to make products in your own home that produce a lot of unrecyclable plastic waste, like toothpaste, for example. Or another thing that my family does is we make our own bread, which sounds like it's really involved, but if you have a sourdough starter, it doesn't take that long. So little things like that, which, I acknowledge take time, and not everyone has that time. If you don't have the time to make things in your own home, doing things like buying in bulk from a co-op where you can refill your own containers is also another thing that we've done. I acknowledge that this is more challenging when you're concerned about the viral transfer. But something else you can do during a time when we can't necessarily be easily going in and out of grocery stores and refilling containers is you can buy in bulk at a warehouse type of store like Costco or Sam's Club. You're buying in bigger packages, and so you're using fewer, smaller packages. Or try to avoid plastic and buy things in cardboard or glass when you can, since those are easier to recycle, because even if a plastic says it's recyclable on the bottom, that's not necessarily the case depending on where you are. So just trying to avoid plastic as much as possible would be another overall recommendation.
Those are some really great ideas, Dr. Brander. And we'll end it there. Thank you so much for joining us today.
Okay, thank you so much.
And thank you, listeners, for catching the second episode of PLASTICAST, the Pacific Northwest Consortium on Plastic podcast. Tune in for monthly episodes as we explore the world of plastic pollution, research, and policy. I'm your host, John Dickens. Have a great week and stay healthy.